Okay. No, no, I so thanks for coming. The morning session. I want to thank Saroj. He's been a friend since 6 days, which is a long time ago. You can hear me at the back, or do I have? Okay. The microphone still okay. So I just hear my echo. Uh, so I'm very happy to be here. I changed the title of my talk just because I wanted to make it a little bit more uh, thinking outside the box kind of a discussion rather than going into depth and details of some of the platforms we have implemented. Let me turn off my, I'm hearing my own voice here in my computer and it's driving me crazy. Okay. All right. So I'll do that. Just give me 1 second. Okay, now it's working. All right. Okay. So what I'm going to do is show you a journey through the world of what I call flexible bioelectronics, which is, I would say, looking at electronics maybe a few years down the line, right? So you're all looking at circuits and systems that you are using hard substrates like silicon, and most of the companies here, we make a lot of very nice silicon chips. Uh, and yeah, I think, that I think is from here. Okay, let me try one more time. 1 second. Another port. Okay. Leave it like be very gentle about it. Okay. So as I said, I'm going to switch gears and talk a little bit more about flexible bioelectronics. So before I go there, I just want to give you a brief background about where I come from. I'm from Tufts University. If you haven't heard the name of the university, it's okay. It's not very well known, but it's one of those very small or mid-sized universities in the Boston area. It's actually quite old, since 1852. So we've been around since the time of Jhansi Ki Rani, right? So that old. So give you a perspective, it's older than even MIT, and a lot of great things have happened at Tufts. It's a university which has, I could go into the history. I love sort of finding out hidden stories and gems about the university and the people who work there. And there is a storied history on radio, wireless, from Tufts. And just to give you a little background, there was in 1916 to 1930, this was a time when people were still trying to figure out what to do with the wireless communication, that they had invented, the radio. And they were predominantly using it for transmitting Morse codes for messages, right, to communicate across the Atlantic. Uh, so the first time the broadcast ever for entertainment that you could actually do radio sounds and music, that was the first. That thought never appeared until Vannevar Bush, who is the, if you know, National Science Foundation, a big funding agency in the United States, and he was also an academic dean at MIT, spent many years there, but he was actually a Tufts graduate student. He did his bachelor's and master's, decided that, he will set up a tower at Tufts University, and he broadcast, and then without the army and the military knowing about it, uh, music for 3 hours continuous. And somehow people in New York and the army people found out that they were getting all these weird messages rather than, and so they shut it down. But the point was that, a lot of really interesting pioneering things happen at a university. The CAT scan that we typically go to the hospital to get is actually was invented by a faculty at Tufts. He won the Nobel Prize for that. So there's a lot of very interesting stories behind the university. And one key thing that goes through all of this is that university is also very supportive of what I call interdisciplinary research. So when you work on a problem, I was mentioning briefly yesterday that it's useful to cross over the disciplines and try to figure out challenges that typically lie at the boundaries of various disciplines, right? And so the university is very supportive of doing interdisciplinary research. And I think the next new inventions and new discoveries and things that will change the world are happening at the boundaries of various disciplines. And my talk kind of hinges on some of that, and it sort of takes the tough spirit of interdisciplinary research and sort of brings it brings it to you. So let me just show you the vision in my group, so you can see the slide. Okay. So in my own research group, I started off as doing circuits. I did a lot of analog mixed signal circuits. I really enjoy doing that. I still sort of enjoy. But then I realized that I need to solve some fundamental problems, right? I mean, I'm designing circuits, but it has to be geared towards some interesting problem. So this is a vision that I wouldn't say I started with this vision in mind. It's just a vision that I sort of crystallized over the years, and it just looks like this, right? And so of course there are many interesting problems in the world to solve, but one of the things that I'm looking at is saying whatever we do, we need to be able to understand our health, right? So there are a lot of factors that affect how our health is, right? So diabetes, people have asthma. If you live in a polluted city, you have a lot of lung related disorders. And if you look at medicine today, the way people practice, it's still considered sort of based on heuristics. It's not really driven by pure evidence. So people know how to treat a certain symptom, but they don't know how to treat the disease itself, how to cure it, how to not have it. And so one of the things we want to do is see if electrical engineers, we are not doctors and scientists, can we make an impact? And the answer is in front of you. You already knew this. We talk about Internet of Things. We talk about Internet of Everything, right? So these are devices that are connected. They actually track stuff in real time. So why not try to use that for health, right? At the same time, if environment is affecting our health, we want to know what's in the environment that the water we drink or the food we eat, can we monitor that and see if there is something in there and then link the two, right? So now you have a cause and effect. You're closing the loop around your body, right? So you know that I'm breathing this air, I'm drinking this water, and 20 years down the line, I'm getting cancer. And now you can trace it back to the root cause. So this is also feeding into this whole new narrative of big data, right? So you want to generate all this data. To do all of that, you need all this information, right? And sort of links very well. So I'm basically saying that there is a lot of opportunities out there in this area of smart and connected health, right? And to do that, you have to be able to figure out things about your environment and health. So what we would typically like to monitor in the environment are things like how's our air, water, food and drinks. I mean, I also want to know other things, your occupation, what kind of work you're doing. And so the work also impacts, that's also your environment. Social stresses are also considered environmental factors that affect your health, physical activity, sleep, and so on. So we want to be able to make sensors. We want to be able to understand these different parameters in the environment. At the same time, we want to monitor our health. And so what I mean by that, so many different things. I just mentioned diabetes, asthma, neurological disorders. And eventually we want to go into a pathway where we can do some early screening, right? So even before you get cancer, you want to know whether you are susceptible to it, whether you are going to get it, right? So platforms and devices that allow us to get early screening of these is quite useful. So does it make sense, right? So everyone with me on this motivation? I think that's probably the key here, is to connect the health and environment into and trying to understand how we can make platforms. So this is our goal in my group, is to basically, I was a circuits person, but we decided that to do this, we need a holistic approach. And that involves understanding and innovating on the materials level, trying to build devices on our own, trying to do circuits and systems. So if I may ask, what would you say in terms of making a platform, one of the key requirements there is here? Anyone? If I were to make a sensor that monitors my health, what would you think would be the most important criteria? What that sensor or platform needs to do? Pardon me? Go for it. Control brain activity. That will be controlling some aspect of my health, right? So I'm looking at a very general, very simple, pardon me? Spinal activity. Well, if I want to monitor my lung health, maybe I don't need to monitor spinal activity. In general, I'm talking about like the physical sort of parameters. It needs to be something that you can wear on your body, right? Something that's flexible. It could be implanted. You're going one step further, very futuristic. That's great, but you're looking at things that are basically flexible, something that interacts with you on a daily basis, right? So the reason for flexible bioelectronics, this is basically where I'm going, is the key requirement in all these platforms. If you're monitoring your health or in your environment, you can't have things that are bulky that you can put on the body. If you want to monitor your ECG, your biopotentials, or your EEG or brain activity, you can't put a hard substrate in the brain. You want to keep something flexible. So the point I was trying to make is the most key requirement in all these platforms is to have something flexible that is compliant with how we are, which is flexible and stretchable. And so even if we are making platforms and devices and systems, they need to be inherently, the materials need to be flexible, right? So that brings me to some of the examples out there. And of course, I'm just capturing. So this is trying to give you a sort of sense of what's being created, what's being invented by some of the leading researchers in the world, right? And so I know you're working on some exciting things for your projects. Ask yourself a very fundamental question, right? If you're monitoring human body, things need to be flexible. They have to be on the body. So for example, this is just, there are many slides I use for sort of motivating this. But the first one, for example, on the left, this was a paper published in a very respected journal, Nature, on doing sweat sensing. You know how people have Apple Watch, they monitor, or your smartwatch, they monitor what? Heart rate. And what else do they monitor? Step count, right? But if you want to monitor cancer, is that going to be enough? No, it's not going to be enough. Why? Because the information is inside your body, it's not really contained just in the signals. So you have to have access inside, right? So the next generation of smartwatch should do what? Monitor the chemicals inside your body. Not enough just to monitor your potential. So this is saying sweat. You sweat all the time, right? And so if you're an athlete who wants to improve their performance, the sweat contains a lot of useful information. There are ions in there. There is a lot of biomarkers. You use the word biomarkers. These are markers of your health. So we call them biomarkers, right? There are a lot of biomarkers in sweat that if you monitor and track, you will know how well you're doing in any given exercise. But it can also be used to monitor in general health. So now if you want to monitor sweat, you can't have a big instrument sitting and connected to you and you're running on a treadmill. It's not going to work, right? There is a need for flexible bioelectronics, something that sort of connects to you in very intimate way so that they can get information. So this is basically showing Ali Javed's work, very interesting. It has a lot of electrochemical sensors. I'll show you some of our work also, but I'm basically trying to motivate. And these are sensors. You can see they're flexible. If I have a pointer, I can use that, but that's okay. So there are flexible sensors on this side, and then there's a flexible circuit board, right? That basically connects. Now you can imagine you're doing circuit design. You want a problem to solve. Why don't we come up and figure out a way to integrate this large circuit board with a lot of function in there? There is a readout. You're sensing information from the sensors. You're digitizing this. You're communicating this wirelessly. Try to integrate them into a small packaging platform. That would help. I don't want to go over all the others. This is showing some other work that has a lot of very interesting features. And the reason why I'm showing you this is because it still leverages existing process. I'm not showing you platforms that are truly flexible, even at the transistor level. These are still using chips. These are still using the platforms that you do, but they're packaged very interestingly enough that they can flex and stretch, right? So you can basically put them on the body and sort of track and monitor. The last one here is even more interesting. Here, even the transistors are flexible. So they start with the same silicon, the same transistor that's used in a CMOS process. They thin it down, do some sort of post-processing, and after thinning it down, they figured out a way to transfer that thin transistor on a flexible substrate. Okay? And one interesting property happens. If you're a mechanical engineer, you know this. Your bulk material is very stiff, and if you keep thinning it down, its mechanical properties change. There is a relationship which is sort of third order where the stiffness changes and your your material becomes more elastic. Even silicon is stretchable if you go down to few less than a micron, less than a couple of microns. Who would have thought? Silicon looks very brittle, sand is very brittle, but if you thin it down, it can be stretched. And so John Rogers created this very interesting ideas where he takes a commercial silicon process, thins it down, and makes lots of very interesting devices doing that. So what he did here was he made an inductive coil for doing wireless coupling. There are transistors, there are sensors, all those things. There is no more USB in mine. That's fine. It's okay. I'm just going to exactly. So the last two are basically something from our work. So we looked at some niche problems. So I was at I'm an electrical engineer. I spent a year at Harvard, MIT looking for problems. All I did was listen, figure out if there is something I can help with. And so the first problem that came to me was, let's say I want to monitor wounds, and I'll give you a reason why we are doing this. So this is a smart, flexible bandage that can monitor how your wound is healing, and then deliver treatment if your wound is not healing well, right? Closing the loop, sense and then react in an appropriate way. And there are some very interesting platforms based on that I'll show later. Okay, so I'm going to basically go through a few of these, and I can go through them very quickly. If you have any questions, I like just raise your hands. Say, I have a question here, and please stop me, and I'll be glad to go over that. Okay, so I'm going to start with some guiding principles for how we do things, right? So there is the World Health Organization came up with this term, and it's probably known by some of you. Is a word called assured diagnostics. Let me move this out. So this is a term that was coined by the World Health Organization, and what it stands for is that if you're making something, at least have these guiding factors. It should be affordable, right? So if you're doing flexible electronics, and if you need a $1 billion equipment to make all of those platforms, not useful. It's not going to scale well with the developing world. It's not going to be that useful around. So try to make it affordable. If you're sensing something, make it sensitive enough. I'm not saying sensitive to the extent that you're overdoing it. Sensitive enough for the application at hand. Make sure it's specific. It's not generating garbage. It's generating data that's relevant. Should be user friendly. Not everyone knows how to use some of these diagnostic tools or this equipment. Smartwatch, do you do anything? No, right? It just tells you everything in the app. You don't know what's happening inside. That's user friendly, right? So it's important to keep in mind that if you're doing something, that it is basically user friendly. It's rapid. And the last two words I like the most, equipment free. Now I am, basically I'm saying if you're diagnosing someone, don't expect them to buy a big instrument to process and do the readout. See if we can do something that doesn't require them to put a lot of infrastructure. So if you're doing blood analysis, I don't want to put a big instrument that does all the blood pathology to the extent that it becomes too expensive. Something that's simple and equipment free and delivered. Something that can be, for me, delivered means it should be delivered at any time, anywhere, right? So these are just basically guiding principles I wrote down in some detail what it means to us as a group when we're doing research. So if you're doing some platforms, make sure that you're not using any expensive materials. If use locally sourced materials that you already have in front of you. If you're trying to make it low cost and delivered, you have to make sure that you are able to fabricate those devices or platforms. Not just any time, like you're doing it in the developed world and then transporting it to places where we need, but you should be able to fabricate them locally wherever they are needed. So it's what I call do-it-yourself kind of diagnostic. And when I say equipment free, I basically mean no expensive instrumentation needed, right? So that's so you can use existing consumer electronic devices. Your phone has a lot of function today that if you just knew how to leverage that for applications that you have not thought of, like in medical diagnostics, that's what I call equipment free, right? So you're leveraging the power and the processing of the platforms you have in front of you to do interesting things. Ease of use. So if you're making something, it should be easy to use. Direct sample to answer. If you're doing point of care diagnostics, you drop in a sample. Don't expect people to do the experiments over and over again so that there are errors. Make sure that they basically come out on the other end and you get the data you need right away. Okay. And at the end of the day, you have to also worry about the end of cycle, right? So end of cycle thoughts don't appear in product development, but it's very important. So if you're making a platform or a device, what happens after you're done? So if you're making a diagnostic device, is it toxic? Is it environmentally friendly? You need to dispose of. So I'm using this to show you sort of the guiding principles behind. We don't always meet all of them at all the time in all the projects, but this is basically something that we go. So let me show you the first work, and this basically sort of fits into this. So this is using, I want to make a diagnostic device. I want to make it cheap. What's the cheapest thing you can see in front of you? Paper, right? Paper is very cheap. Can we make interesting devices out of paper as a substrate, right? So there's a lot of effort by a very well-known chemist in Harvard, George Whiteside. You know, he's a chemist, but he does idea of interdisciplinary research. He does a lot of very interesting things. And he asked this very interesting question: why can't I do this with this material? You know, what can I do to engineer this material to give me the function I need, right? So paper is one of those very interesting platforms. So paper as a smart substrate. So not silicon, paper as a substrate, right? So I'm thinking, so what is it that paper has that we can leverage, right? Well, you can print anything on paper, right? So right now you're printing inks, right? So what if that ink was smart? So if your ink had a function associated with it, you can actually print that too. And the reason why it is used it's easy to do that is because printing has been around for centuries, and we know how to write with inks. So all you have to do is, if you're making something on paper, you make a smart ink, right? So if you can make an ink, then you know, for whatever function you need, then you can basically use leverage printing as a way to get your smart functional devices. This is very important. I've been talking about biology, monitoring human health. Biology is wet, right? So if you're monitoring blood, or if you're monitoring saliva, or you're monitoring any biomarkers, and they're inherently wet. So, and how do you do wet chemistry on paper? And to be able to do that, if you look at the most common thing most chemist or any lab technician would do with any biological is they would mix with a certain ratio. They will wait a certain time. Then they will purify that sample, mix it with other reagents. This process is what is called fluid handling or sample handling. That requires a way to transport liquid. That's the most fundamental, and mix. So mixing and transport is very easy on paper. You don't even need a pump or a valve. Why? You know, when you write with a ink pen on paper, it blots, right? You have a natural capillary wicking action on paper that takes an analyte from one end to the other. If you can only control this process, now again, you're using engineering skills to figure out, okay, what's the fundamental way the fluid is getting transported on paper? Can I do something? Can I introduce reagents while during this transport process to modify that? And so that's basically, paper is very useful because paper has a natural zero power or power-free fluidic transport. So you can do extraction, mixing, dilution without pumps and valves. All you need the most important challenge was to be able to, if I just put paper in liquid, it'll all wet. If I want to make it go somewhere, I have to basically pattern a microfluidic channel, right? So how do I get a channel on paper? You have to be basically able to print regions that are what I call hydrophobic, that don't like water, but and then regions that are hydrophilic, that like water. So how do you get these two regions? Well, you can do something called wax printing. Your ink is a wax. You print where you don't want water to flow, you would print with wax. Where you want the liquid to flow, you will not print with wax. So you can basically get channels. This very simple observation led to fundamentally very interesting devices. So this is George Whiteside's work. And it's not possible actually. It is so powerful that it's not even possible today with existing approaches to get a device like this. This is a three-dimensional microfluidic channel on a single piece of paper. There are almost 3 different fluids that are flowing in this, and they're flowing at different levels inside a single piece of paper. And the way you have done that is basically patterning hydrophilic and hydrophobic channels, right? And they don't even mix. You can see different colored dyes for representation occupy different sort of channels in there. Very simple process. You just wax print on paper, design where the fluid is flowing. And this is basically showing you the power of sort of engineering, so frugal engineering, right? You start with paper as a substrate and you say, I can actually do interesting things, even better than what is possible with some of these other substrates. So just a little bit out of the box thinking, asking a very important question as to, can I get something fundamentally more interesting than what people are doing, will serve you a long way. It's very environmentally friendly. If you're doing diagnostics, once you're done, you don't want someone's blood sample contaminating someone else's, right? So, and if you are in Africa or places where there's Ebola spreading and you have a diagnostic device, what do? You have to dispose of that, right? So the most bio-friendly way of disposable, getting disposed is just incineration, just light a fire. So paper will just burn and you will just basically, so it's a very environmentally friendly way of disposing sort of biotoxic waste. So I don't want to go over sort of, this is just basically a picture slide trying to give you a vision. And this is from Whiteside's group again, and he's showing you all kinds of different things you can do. The only thing I'm showing here is you can see you can do dilution. You can dilute precisely, you can mix precisely, and so on. So what did we do with this? We actually made a platform that would allow us to do electrochemical sensing. Okay, so electrochemical sensing of some analytes in any biological fluid. So it could be blood serum. So you take blood and you extract the serum part, remove all the cells. There's a lot of biomarkers. We also work with saliva. Saliva is a interesting rich source of biomarkers. And we basically wanted to see if we can do some of the biomarker sensing on paper-based platforms, like the one I showed, for some early screening or for some sensing kind of applications. And there is an interesting need for merging electronics with the sensor platforms. And so you will, at least I'll be able to show you some of that. Okay, so this is just a background slide that if you're doing some sensing, there are 2 ways of sensing, simple 2 ways. Either, how do you know something is there? There's a color change, right? Some way. So this is called transduction, right? So there's a biomarker you want to see, you want to convert that biomarker into a signal you can read. And that process is transducing the signal. And so when you transduce a signal, you can transduce the signal in a color forms where you can see color change, or you can do electrical change. There is a change in current, or there is a change in voltage, or there is a change in impedance, right? So you have to go through that process. And so there are colorimetric where there is color assays, and there are electrochemical, which is sort of blending chemistry with electricity. So what we found was when we started doing this work a few years after everyone was trying to do this, was there was a problem with paper diagnostics. Again, this is asking the same fundamental question. There must be something to address this. And one of the questions we had was, there was too much variability from sensor to sensor. You made one sensor, you made another sensor, you made a third sensor. They were not all the same. And the reason was that the paper is a rough substrate. So if you're making any electrochemical sensor, you have electrodes that are different size. The surface topology is different. If you have different surface areas, you get different values of the signals that you're transducing. Not a good platform if you're doing accurate diagnostics. So we wanted to address that. This is just a basic slide on electrochemical sensing if you're interested. So what we did was we made something called a 3D. Now this is interesting. We made a three-dimensional platform using paper as a substrate. Now how do we do this? This is basically showing the process flow. We start with a paper substrate. Again, this is equipment free. You can do this today in your lab without going to a fancy clean room, right? It's that simple. It's nice pictures, but it's basically many and different ways of doing this. So you start with paper, you put an adhesive, a two-sided adhesive tape, and you basically fold these on top of one another, right? And then you basically use some kind of a cutting technique. So there's a laser cutter, or you can also use just a blade, right? Whatever you have, you cut out and open the surface on the top. And I'll tell you why we are doing this, okay? And and then what you do is you do something called screen printing. So I showed you how paper is very good for writing things because you can just print. You can use that, just load different inks. So here we have three different inks we use. So one of the ink is a carbon ink, it's a conductive carbon ink. And this carbon ink you can coat it with different sensing chemistries, right? So this carbon ink can be loaded with enzymes that are sensing glucose. The glucose test strip, most diabetes people who want to monitor use, they're basically running on this scheme where the blood is extracted. It's one of the paper fluidic devices. It extracts the blood. It takes you to a region where there is something called a glucose oxidase enzyme sitting on a carbon electrode. And when the glucose comes in contact with that, goes some few electrochemical reactions, there is an exchange of electrons happening with the electrode, and you monitor that current. Okay, so there is sort of a direct relationship between glucose and the current you read out. And that's also implemented using the screen printing technique. So we can use any and different methods to get these electrodes. You can literally write with a pen, load your pen with that ink and do that if you want to do it low throughput, or you can use a fancy inkjet printer to basically load that. So once you get these inks, then what you do is you fold this on top and you get what is called a three-dimensional paper analytic device. And so why three-dimensional? So this is how the device would look. You have an electrode on the bottom, and then you have these wells on the top. And the reason why we did this was, remember, we had a problem. The problem was each sensor was different. We wanted to get a smooth surface. So what we realized was the original paper devices were sensing on the bottom surface. So the fluid was flowing into the paper, and the electrodes surface that was interacting with the fluid was basically rough, and it was into the paper. But when we print, we get a very smooth surface on the top. So printing is better. So if you can sense on the top electrode surface, you will get a much better response. So this is just basically showing you that you can be creative with simple things and that address very fundamentally interesting problems. And this is basically addressing that, the exact thing that I said. So if you're doing two-dimensional sensing, the electrode surface that faces the liquid is on the bottom side, it's rough. But if you're sensing in a 3D reservoir on the top, the surface is more uniform, and you can basically get much more uniformity, and there is much more. And also, the top electrode, you have access to the electrode itself, so you can functionalize different things. If you want to monitor glucose, you will functionalize glucose oxidase enzyme. If you want to monitor tomorrow lactate, you can functionalize with the lactate oxidase enzyme. So there are different things you can do with these electrodes to sense different. So, but how does the entire platform look like? It's not just that paper substrate. You need some electronics to go with it, right? So again, I was talking about equipment-free readout. I don't think you can do equipment-free readout with a electrochemical sensor. You will have to have some simple, cheap interface module right there, and then it can interact with your cell phone, right? So that will be your final destination. So you need to bridge that. So this is just showing you one way of, if you're a circuit designer, where how you can basically help with flexible sort of diagnostic platform. So this is an example of that. So imagine you have a disposable paper-based sensor. It has these three electrodes, and the three electrodes will connect to an electrode on a disposable tray, a little tiny strip, just like the glucose sensing strip. You put this sensor, you drop a drop of blood or whatever you have on top of the sensor. It goes to a electronic chip. What does the chip have to do? What is it reading? Here the transduction is in current, right? This signals coming in current. So it has to interface. It's a current readout circuit. It's reading basically a current. So it could be a transimpedance amplifier if you've designed that, right? Which takes current and generates the voltage. But it also has to apply a signal to the electrochemical sensing electrode. And the voltages are typically a sawtooth waveform. You change the voltage up and you change the voltage down, you change the voltage up and down, and you read current as it's doing this. And that's basically, so the circuit has to apply a voltage and then read a current. And you have to do a little bit more, but the essence principle is this. You apply some stimulus, you get some signal for most sensors, right? So, and that platform, if you're interacting with electrochemical sensor, is called a potentiostat. Okay, the potentiostat. If you look at the potentiostat today, it's a bulky instrument because it does a lot of different things. But if you're doing just this, you don't need that. You need a small, simple, custom electronic device that can actually do the function you need, and that can be made very small. Right? It's not very complicated. You can connect directly to the phone if you have a port or something. In some cases, you want to transmit wirelessly based on the application, so you might need a wireless transmission module. Last but not the least, you need some kind of power source. Now, if I'm making disposable diagnostic devices, and if I'm using battery for everything, it's not going to fly. That's just too much batteries. What's the power requirement in this? One-time use. Should I use a battery? No, it's a waste, right? So if I'm just taking a measurement, throwing it away, you can use a battery if you're not making a disposable sensor. You can repeat, if you reuse, then you can use a battery. But if you're taking a sensor and just throwing it away, we need to come up with some platform. And that's why I mentioned you have to come up with a power source that will power up this platform, right? So as electrical engineers, you have to think of, okay, how am I going to get a disposable power source? Just have enough power for a short amount of time and just forget, right? And so I'll show you some. There are many ways. People have used your saliva, your spit to power these sensors. Just spit on it, and there is a little bit, there's enough power in there is enough chemistry in your spit to actually power up your sensor. We haven't done that, but I'm just basically saying there are many ways of powering up these devices. All you need is a power for a single time use, and you can use them through. So if you're a circuit buff, you can appreciate very simple op-amp that goes in. This circuit is actually the simplest circuit you can implement. It's an op-amp with a resistance and feedback, a transimpedance amplifier, another control op-amp that actually applies that sawtooth waveform I was telling you, right? A collection of two op-amps and you're done. That's all this is, this potentiostat. After that, you need a digitizer. You need a ADC, you need all of that, but I'm showing you what a student implemented just to get this to work. And then you need a nice interface, right? I mean, just to basically on the receiver side, once you transmit over the phone, wirelessly on the other side, someone has to make an interface. So this is showing you that you can actually make an interface. So this is an actual chip with an actual device, right? That does, and interestingly, this can sense 3 things at a time. There are 3 different working electrodes. Each one functionalizes with 3 different things. So you don't just monitor glucose, you can monitor dopamine, which is a marker of your brain health. It also appears not just in brain, but it also appears in blood. So you can monitor dopamine, you can monitor glucose, you can monitor pH. Tomorrow you can monitor anything. This platform is universal. Cheap, low cost, disposable chemical sensor to monitor your health, right? So that you can definitely see. So these are the different things we sense. Go ahead. Go ahead. Rest part you also throw, because this is, if you're just doing, well, for us, we want to reuse. We don't want to keep throwing. So if you use a battery, then you will use the tray. You just throw away the disposable sensor, right? So you just throw away the sensor. Otherwise, you just put the second sensor on top. This chip is the CMOS part, the one that is away from the conventional method is still done. Electronics is still done, because this does not require you to do flexible electronics also, right? That's an overkill. You all need is a device which, the reason why this is flexible is because paper is much better in handling fluids and patterning and doing the device fabrication than going into the clean room and doing this process, multi-step metallization and all that. Why do you need all that when you can do it? Yeah. Oh, I mean, I agree, but this is a tiny grain of silicon, right? I mean, if you're a CMOS, you want to, this is not toxic. There is nothing here. The battery is toxic. The battery is the problem in disposable, not the electronics. One grain of sand should. You can just use a Bluetooth or any other transmitter right now to basically get this to work, right? So this is basically just off the shelf. We don't reinvent the wheel on everything. You can just basically. I think the only thing my student Mira she made was this potentiostat, just because that was something that she wanted to interface with. So you can monitor many different things. This is showing you that you can monitor pH. You know, blood acidosis is very important for people who have kidney or any of those ailments. So you have a disposable sensor for monitoring blood. You can monitor glucose. So you know what kind of readings we take. This is what is called a chronoamperogram. You apply a step voltage and you measure how the current changes. Right. So if you're an electrical engineer, you know the stimulus, you know your readout. You can design a circuit. This is how the current waveform looks if you do cyclic voltammogram, the one where you change the voltage up and down. Right. Okay. So just giving you a flavor for sort of the process of making diagnostics, I'm just going to go show you some more. We also made some very interesting platforms, you know, like making very high surface area electrodes on paper. So people think you are using cheap, low cost substrates. You cannot do things that we can do with a clean room, like growing nanowires on silicon. Why people do that? For batteries, high surface area electrodes, you can only grow nanowires on silicon because there's a clean room process. You do a lot of fancy steps and you get. Well, this is a process that can grow nanowires on paper substrate, vertical nanowires on paper substrate, arbitrary any nanowires using a solution process, right? So you take your paper, coat it with some metal, there is a process there, but you can actually grow nanowires controllably on paper substrates. So what can we do with that? So these are showing you some nanowires. You can go nickel nanowires, copper nanowires, platinum nanowires. Go ahead. Pardon me? So we don't print nanowires. So this is the electrode deposition process. So I can go into more detail of the process. So previous you ink, you just write because those are micron size wires. Here, these are nanowires that we grow through a template. So there is. Yes. Yeah, the variation does matter. You have to calibrate. So that's the other side of things that you have to worry about. You know, when you print, how repeatable the printing process is. So all our work, when we get the sensor, we do something called a 2-point calibration. So before we use the sensor, we calibrate one time. After you finish all the reading, you actually calibrate one more time just to see. And the calibration process, this is a process you have to basically do. Or if your printing process is repeatable, then you don't have to calibrate the second one. You can just do initial calibration. It will basically work 1-point calibration in the beginning, and you should basically be able to address some of these variables. So this is a process which basically uses something called anodic aluminum oxide template. So this is a, you know, if you take aluminum, I'm not saying you should do that and you'll get this, but the process is as easy as taking aluminum and putting it in acid at a controlled temperature and a bath. It naturally forms these pores, you know, and the spores are nanometer size. Okay, so this is very interesting. If you control the process, you can get different size pores. Now you can use those pores as a template to grow nanowires on paper substrate. And so we did that. We grew a lot of different nanowires. And what can we do with this? I just basically put a one slide to sort of give you. We can do something called ECG recording. You want to do ECG, right? Monitor health. So what do they do for ECG recording? Put a gel, right? Put it. If I want to monitor for 24 hours, the gel is not good on your skin, right? It will dry up and it will peel off and you'll have all kinds of issues. Well, here, when you do nanowire electrode on paper, you have a brush, and the brush will always rub against your chest or your body, even if you're moving. So now you have a dry ECG electrode, right? So you have an electrode that can monitor ECG signal without really using a gel. You're addressing a very interesting fundamental problem. This is a cheap nanowire brush on paper. You can dispose them off. Probably not as cheap as the gel electrode that you get, but it's definitely quite cheap. Right? So the point is, you can do really interesting things that people have not been able to do when you just sort of look a little bit outside the box and see that, you know, there are ways you can get some of the interesting things. We did make a battery. At the end of the day, you know, I was saying there's a paper-based battery on the other end. So we did make a battery for a different application. This is a paper-based battery. We use these nanowires on paper. when you have these nanowires, you have a large surface area. If you have a large surface area, all you need, what is a battery? 2 electrodes in a acid bath. If you want to use one-time disposable paper source, all you need is a source of acid on those 2 electrodes, right? So here, this is an example showing zinc and platinum nanowires. You can also use zinc and copper. Platinum is expensive, I understand. And you can basically put a little filter paper, a little membrane on one of the electrodes, all low cost. And in our case, we basically used our own source of acid, whatever is there in our body. So the most acidic source I'm showing is our gastric acid, the stomach acid, right? So of course, not for this application, for another application, we basically tried this. But you can also use, even sweat has a pH of around 6, right? So there is different sources of biological fluid, you will get some power source. But this is showing you the really nice performance. It generates almost a volt, milliwatts of power from a disposable paper source, a paper-based battery. So you can do even more advanced things when you use very simple sort of platforms to do this. So any questions so far on paper-based, right? So I'm basically showing you what are the different possibilities. So I don't know how much time I have. I can show you 1 or 2 projects, I think. So 10, 15. Okay, so what I'll do is I'll skip this. I'm just going to show you the journey, right? So that was electrochemical. We can do colorimetric, where the signal transduction is not from biomarker to electrical signal, but it is color change. It causes change in color. So I'm going to skip this. And so this is basically showing one of the application. I'm just going to show you this motivation slide. And this was to do saliva diagnostics, so, and this is a problem that's very common in Asia and Middle East. This is an infection with a so when here, most of us actually do have bacteria in our gut, right? I mean, that's natural to have. But one particular bacterial infection is known to cause polyps. The polyps will become cancerous and you get stomach cancer or colon cancer. So one of those is H. pylori, Helicobacter pylori. It's a gram-negative bacterium that actually is there in your gut. And what it does is basically, if it's there for a long time, it's going to do its thing. It's going to break your intestinal walls. It's going to change the thing there, and you basically start developing polyps. So it'll be nice if you knew you have this infection. How would you do that? You go to a doctor, and the only thing doctor will say is he'll put a tube inside your stomach, into your mouth, into your gut, and try to extract out the acid. You're not going to say, I don't care if I have H. pylori, I'm not going to do that test, right? Have to find a non-invasive way if you're doing early screening, right? So apparently it happens that saliva is a very good surrogate for gastric juice or blood sample for especially this particular, not for everything, but this particular application there is. So what happens is this bacteria, when it's in your gut, it doesn't like the acidic environment. So what it does, it releases this enzyme called urease. And the urease will then basically make that area where the bacteria is a little bit more alkaline, just so it can survive in your stomach. And the way it does that, it generates this ammonium and CO2, this carbon, and that high levels of ammonium and CO2 in a gastric juice correlates very strongly with the one in saliva. So what we did was we made this colorimetric sensor, which can sense ammonium and CO2, dissolved CO2, not the one that we breathe in, dissolved CO2 in your saliva. And so this is again a screen printing technique to make these electrodes, and you can basically do very nice measurements, showing you that these 4 different colorimetric dyes, you can basically sense the level of CO2 and ammonia. It's very precise. The levels we can detect were parts per million. And you know what we use for readout? Your phone. So we look at the color change with the phone. We take a picture of that color change. We upload to the cloud. It finds out how much each channel, red, green, and blue changed. So you get some sort of quantified readout from. So this is a completely equipment-free, cheap, disposable early screening device for monitoring whether you have H. pylori infection. Okay, so I'm going to spend the last 10 minutes on a very interesting project, the other project in my group that I showed you a nice picture for, something called the flexible bandages. And so this is a problem that was presented to me. Of course, we can do a lot of different things with flexible bioelectronics, but as I spent my 6 months at the medical school, I realized that one of the problems they were trying to do was trying to heal wounds. If you're diabetic, and if you know someone with diabetes, there is one problem that people have is when they get a wound, it doesn't heal, right? And the reason why it doesn't heal is because your pathology is all bad in the body. But one of the main reasons is there is something called diabetic neuropathy. You don't really sense what's going on in the extreme. So you basically develop these wounds and you ignore because you just don't feel it. The reason why we pay attention to any little cut is because we feel the pain, we see the blood flowing, and we want to do something about it. But with diabetes, some people, they will not even feel that the wound is there. And in most of these cases, the wounds get really infected, and you basically have to amputate the leg. So most of diabetic foot ulcers, the feet are amputated. The point I'm trying to make is that there is a problem there, right? So we need to heal these wounds. There is no real standard way of healing. The doctors will basically prescribe some antibiotics, some bandage, but it doesn't really heal. And there is a fundamental problem right there. It's a treasure trove of problems for an engineer to solve. The reason being, all they do is bandage up, give you a tablet or put some ointment. It is not treating how the wound is supposed to heal. The wound has a certain way of healing. There are different phases of healing. In each phase, it releases some growth factors, it consumes some reagents, and then it basically goes this process. There's a vascularization of tissues, nerves start to form. If you do not promote the healing process at the right time, it will probably never heal fully. And so there is an interesting problem right here to basically look at. First of all, you want to do the first minimum viable product, right? The first thing is, can we at least monitor infection? Right? Second, can I treat wound? Third, can I do something more? Can I actually heal faster? There are many levels of things that you can do. So here you definitely need flexible substrates. You can't put a hard silicon on that wound. It has to be all flexible. So this is, I'm just basically showing you a vision slide, and it does have a real device. So this is an actual smart bandage. And this was a project, as I said, when I started at Harvard Medical School, and then with Tufts and Purdue. And this is our actual real bandage that we implemented. So it's a period of 6 or 7 years. We did this work making sensors, making drug delivery platforms, and also basically integrating the two together. This is showing you a bandage that actually is less than 2 millimeters thick. It has sensors in it, which can sense pH, inflammation, different things in the wound. And then it has these little tiny drug particles. Okay, and these tiny drug particles, if you heat them up a little, it will release drug. If you stop heating, it will not release. So now you have a controlled mechanism to release drugs from this bandage. It's not releasing all the time. It's not as if you put an ointment and it just goes in. It only releases. You remember I told you just a few minutes ago that in a healing process, you want to release growth factors during some time. You want to release some other reagents during some other time. So you need this control, modulation of drug release or drug profile into the wound. That will be the most optimal way of healing wounds, right? Because you really understand how the wounds are healing and you're basically delivering precise treatment. So this is basically showing you some sensor platforms. It senses different ions and different analytes in the wound, and then it releases drugs, right? And it closes the loop. It senses and releases. Nobody had done that before. Engineers, we do it all the time. We never implement or sell anything that doesn't have a controlled closed loop feedback system, right? That robot that Professor Behra showed yesterday, there is always a loop, right? You watch what the robot is doing and then you feedback the command. Why is medicine not doing that? Why is not monitoring you on a continuous regular basis, then delivering your drug precisely at the right time? That's precision medicine. We've been doing this for everything else. Medicine needs to, essentially what I'm saying is engineers have a lot of opportunities there to basically look at this and say, well, I can do this better. I can actually even do the first minimum thing really better than what they're already doing, right? So this is a flexible smart bandage platform. I'm just going to show you a couple of slides. The first platform was made by an undergraduate student just like you. He was a sophomore, basically second year, and he decided that he wanted to spend the summer in my lab. And he said, I want to monitor something in the wound. I said, the most important thing you want to monitor is wound oxygenation. Is it getting enough oxygen, right? I mean, some wounds that don't heal, there's not enough oxygen. So he made this flexible oxygen sensor, just like the other sensor that I showed you. On paper substrate, he He made a zinc ink. You can buy that actually if you don't want to know, if you don't know how to make it. He coated a silver ink. He made 2 electrodes. He put a little saturated filter paper on top, which had the electrolyte necessary. You need an electrolyte for electrochemical sense, and a thin membrane. You can use any membrane. He used a thin silicone membrane. And it just so happens that the oxidation at the zinc and the reduction of the silver cathode actually is proportional. The current that you generate at that electrode is proportional to the oxygen concentration. Right? So now you have a sensor, you have the potentiostat that you may have built, or you can use off the shelf. And if you look at this, this is actually a galvanic sensor, which means it actually has a voltage there. You don't really need a power source. If you don't want to monitor current, the voltage level itself is proportional to oxygen concentration. So it's actually a zero powered sensor if you can just monitor the voltage. But the more accurate way of monitoring is chronoamperometry, where you apply the voltage and you monitor the current in this case. But anyway, so this is what that undergraduate student did. I mean, he basically took this sensor. This is what he implemented: a flexible bandage, nice circuit board. On one side, he 3D printed this bandage, flexible, stretchable bandage. Made a little cavity, put the board inside. The sensor is on the lower surface that interfaces with the wound. Was never done before. Just monitoring wound oxygen wirelessly was not done. Now you can basically get how the wound is healing on your phone wirelessly while the person is walking around and knows whether there is enough oxygenation in the wound. So you can visualize, you can monitor many different things, right? It's not just monitoring one marker. You can monitor. All these are very easy to do and implement in a lab, right? It doesn't really take a lot of expertise. So this is showing you oxygen. Oxygen is a very hard thing to measure. Okay, so if you want to know why it's hard, I explain it basically oxidizes everything, and it's a very flammable reaction. So if you want to measure in the lab, and you say I want to bring an oxygen cylinder and I'll change oxygen concentration, you have to go through a lot of rules and regulations before you can do those measurements. So you do the best you can. So we monitor different ways of getting oxygen, but the reading matches with whatever standard meter that's out there. So this is a drug releasing particle. So this is basically showing you the same platform. So I was telling you how this is a bandage that doesn't sense but delivers drug. So it does the opposite. It basically is doing the other way. So here this is a thin hydrogel. Hydrogel is basically like a jello, right? It's very soft, moist. Wound likes it, so you can't put a bandage. It's a very thin, so it's very nice if you're coating the wound with something hydrogel. This hydrogel has these thin, small particles that have drugs in them. And when you heat this hydrogel a little bit, it basically releases drug, as I was mentioning. Again, this was using the same 3D printed, same electronics kind of platform, made this screen printed these coils, these heat electrical resistors, pass current through them and heat, right? This is a resistive heater, and it locally heats up this hydrogel and then releases drug. And you can see the drug release profile changes. And this is showing you that you can close the loop, right? So I don't want to, so you can send something and you can deliver. And these are just showing you lots of very interesting data. And the only data I really want to show is two. So one of the data that I want to show is this. These are the particles, how they look, right? So when you are at room temperature, these beads look like they are swollen, right? And they are translucent, they're hydrophilic. Water comes in them, right? They like to absorb things. You heat them up a little bit, it becomes hydrophobic, it releases, oozes out all the drugs from it. So you just have to, it's a temperature-based modulation. And what you can do is you can actually time modulate this waveform to decide how much drug to deliver. So there is a baseline release that always happens, but if you want to change the drug profile, you can heat for a short amount of time and stop, it will increase the drug profile. It will release the drug. So now you have control over the dosage of drug. It's not just one pill taken at one time, you have no control, right? So here you can control each time and in space how much drug is basically going. And this is, of course, I don't have time, but this G, the one on the rightmost part right here, basically shows a result on a closed loop bandage. If you just don't do anything, the pH will keep going down, and there is a bacteria in the wound. This is a wound mimic. This is not a real wound. And then when you activate the bandage with the closed loop feedback, the pH recovers, and you can also visually inspect to see if the bacteria is all dead, right? So there's a bacterial infection in the wound. So this actually delivered a real antibiotic into the wound. So the drug release was antibiotic, and the sensors were pH, just to give you a proof of concept. Okay. And I'm going to show you one more bandage. So this is basically a different kind of bandage using threads. Now let me skip all these slides and just show you some sensing platforms. Okay, so this was another sort of bandage that we did for wound monitoring and delivery. So this is another drug releasing bandage, and this does not use. So the way we implemented previously was we had hydrogel layer, right, thin film. Here, what we did was we actually took individual threads, the textile threads that are the bandage is made of, and each thread has a drug. Okay, so each thread carries a drug. So you have a patch, and these threads are conductive. So these are not, your regular threads. So instead of passing current on a separate electrode surface somewhere, you actually pass a current through these threads. The reason is this is useful is because what you can do is if you can have multiple drugs on individual threads, then you can activate which drug to go release into the platform by just passing current through this. And this is done all wirelessly. This was actually one of the most interesting platforms. So this is actually showing you some results. And I'm just going to show you that if you cycle the voltage, the temperature also cycles. You have to heat that up, right? So you can basically show that you can control. Another interesting thing that happens is you have these threads going. Each thread is carrying a drug, right? So if you want to locally deliver drug on one point, you can basically activate those two threads that intersect at that point. And that will release drug locally exactly in the region where you want. So you have a spatial and temporal control on drug profile. And I'm just going to show you one real study on a mice. So this was a diabetic mice. As I was telling you, diabetes, the wounds don't heal. And the only thing that's interesting here is this and this. This is a control patch. This is a wound. Day 0, day 5, day 10, the wound has not healed. Right? This is the patch with this drug delivery thread, and it delivers what is called vascular endothelial growth factor. It increases vascularization. It promotes the nerves to form, the blood vessels to form, so that it actually heals. But you have to deliver only during a certain phase, the inflammation phase. You can't deliver, otherwise tumors will form. Too much of growth factors. You have to control this profile. And you can see that it heals faster, and you can do some cross-section histology, and you can see that the granularization of the tissue is much better in the wound. But the point I'm trying to make, who solved the problem, is basically some electrical, mechanical, some engineers basically got together and said, don't just deliver growth factors. The reason why this is not in the market is because FDA says it causes tumor in half the population. Well, why does it cause tumor in half the population? Someone needed less, the others needed more. If you just gave one drug that fits all, you're definitely going to have some population that's going to not. So the drugs are failing because there is no precision medicine. And why is no precision? Because engineers are not involved. We're not really looking at, we're not sensing how much the wound needs, how much you can deliver, and so on. So this is sort of basically taking you along the path of thinking outside the box, saying there are other very interesting problems that engineers can solve. And they do not require a lot of really, expertise. Very simple fundamental knowledge that you already gleaned can basically help you do this. So I'll basically stop here, but show you some of the other things you can do. You can do sweat sensing patch. So we did that. You can do gas sensing threads, threads that can monitor pollution. You can also do activity monitoring. If you want to see whether you're sitting or standing, a lot of people are interested in physical activity. You can make threads that change their resistance as you stretch. So this person is sitting and standing. You can characterize that information. You don't need an accelerometer. You can basically put your own shirt is your accelerometer. It's basically telling you which arm you're moving, what you're doing. And one of the students actually even put this threads on a glove. So you can do sign language to text conversion, right? So, all undergrads. These are not graduate student projects. These are undergrad students. They saw the thread. What can you do with it, right? Can you monitor human motion? Can you monitor this? And so these are threads in someone's shoe, and he's monitoring how you walk. People walk differently, right? If you're an older person, there is a problem in the US where elder care is a big cost, right? So one of the things you want to monitor whether someone's falling. So once you fall, there's no point in monitoring whether the person's fall, because you already, before you fall, you want to monitor. So how can you monitor if someone's going to fall? And monitor their gait pattern. If you walk, if there's a slight change in your walking pattern, maybe it's the indicator that there is something cognitively impaired and maybe something's wrong. So you can alert the caregiver. So this is basically just taking you in the direction of using these threads to do some activity monitoring. Okay, I'm going to stop here. Okay. Thank you. Yes.
What is the amount of blood sample required for that paper?
Excellent question. Right. So it really depends on how much is there in that blood to begin with. Right. So it's a probability sampling. Right. If I say that it's 1 part per billion, that means it's very rare in the blood, right? So you must acquire a lot more samples to be able to find that, right? So the amount of analyte you sample from the blood depends. Glucose, on the other hand, is very high in concentration. So a few drops of blood is probably enough. But if you monitor a cancer biomarker, you probably have to take multiple samples. But that's where saliva or other sources of biological fluids which you produce in plenty, you can keep spitting on things. If I can tell you, I can tell you whether you have cancer or not, you will do this exercise, where you will get multiple samples on it. So I think it's a matter of what you're trying to test.
How do we reuse that?
We never reuse. We throw that away. Right. Yeah. That's the reason why we want to use paper. Because if I use anything else, I'll feel guilty, like you're already feeling about paper that I'm throwing away something that's useful, but this is very low cost, it's disposable, that you can basically just throw that away. Okay.
Like you said that you can power that paper with sensor using your saliva. And you also said that you can connect it from Bluetooth sensor and all to connect it with my wireless devices. So is that power generated by your saliva enough to power all these components?
No, it will probably not be enough. So if you really need that, you have to do some power management right there, right? So you have to, that circuitry I didn't show, right? Because I'm assuming there was a battery in that initial platform. But again, whatever you're designing, you can be designing thinking about the fact that this power management block is not supposed to power for a long time. It's a short duration power for 5 minutes, right? So what can I do at the circuit level that will produce steady enough voltage, right? That could be a good engineering challenge to look at, right? So is there something simple? In our case, what we do is we make sure that the circuit that we implemented worked at like 0.5 volts power supply. So some of these op amps that we designed in my group, 0.5 volts, 0.2, we even designed a 0.25 volt op amp. The reason was maybe we can get away with not trying to up convert, trying to work with very low voltages. And since the op amp or all the platforms only have to work for a short amount of time, maybe you can just charge up a capacitor, sort of have a very simple system that let it beacon, so you power up just for that amount of time, and then you can throw away the device. So many different ways of engineering the solution to this problem. But that's a good question, right? You're already thinking ahead what can go there. Yeah, that's true, right? So that's the problem, right? So if you are not going to use all the drugs, the solution was first, doctor just tells you take this drug, right? And then if it's a vascular endothelial or antibiotic, you're taking antibiotics even if there is no infection. That's a big problem already in this country, right? So it's a trade-off you're making, right? So there's not a lot of drug to begin with, but since you're delivering only at the right time, hopefully there's not a lot of drug that is there. You're doing very targeted delivery only when you need it, right? So of course there is an end of cycle kind of consideration there. What do you do if you really did not use the drug? So we haven't really thought about that. But that's a good question. Someone has to pay attention to that. Okay. All right. Thank you.
Thank you so much, sir. for the informative talk. Now, I would like to request Sir Saroj, the convener of the workshop, to kindly present a small token of appreciation to Dr. Samir. Sir, kindly accept it.